Uh, Good evening to everyone. Welcome to the day 14 session, day 13 session on B-cell epitope databases. And we have only one more session on tomorrow. So today let us complete the B-cell epitope databases. Before proceeding to today's session topics, can we recap the last session topics? Do you remember what exactly did we discuss in the last session? We discussed something on T-cell epitope databases, how the T-cell epitopes are predicted. We have seen MHC1 prediction, MHC2 epitopes prediction. Remember that? Now, let me interact by asking you a few questions through both. Now, I just launched the first question. I think it's visible to everyone. Which cellular component is primarily responsible for generating the peptide fragments loaded on two major histocompatibility complex class 1 molecules. So again I'm studying which cellular component is primarily responsible for generating the peptide fragments loaded onto MHC1 molecules. So the choices provided are lysosome, Golgi apparatus, proteasome, endoplasmic reticulum. Yes, in fact, we have 15 participants joined in this session. So this is of course related to immunology, but this is also important in immunoinformatics. I mean, whatever I have shown you, IEDB tool and Raghava web server, we also have specific tools which determine this kind of a score. Whatever the peptides we can able to generate, how many peptides, epitopic peptides are generated from a protein sequence. Okay, this is actually predicted through a score. Okay, now I'm going to freeze. We got 9 votes at present. So the maximum answer for endoplasmic reticulum. No, that is wrong answer. The next answer, proteasome, is the correct answer. You know how exactly it fits? I told you that which cellular component is primarily responsible for generating the peptide fragments. I mean, if any of the virus or bacteria are getting inside, okay, after engulfing the virus or bacteria, okay, through lysosome, okay, of course, through endoplasm, before going to the endoplasmic reticulum, that kind of a virus, whatever, if it has to be degraded, I mean, viral proteins have to be degraded, then the viral proteins have to pass through proteasome. Proteasomal degradation is important. So, through the proteasomal degradation score, one can able to determine the number of epitopes formed from a protein sequence what we are going to paste. Okay. So, like that's what we do, isn't it? We go to the tool and we just copy and paste a viral protein sequence, and we try to generate so many epitopes, and we find which score is maximum. That is the best. That's how we have screened. Everything is okay, but the number of peptides dependent on this. The second question I'm going to launch now. The second question means here in MHC class 1 processing prediction, what does the transporter associated protein transport score evaluate? So I have shown you a mechanism also, and I told you whatever virus or viral proteins getting inside. Okay, we also have something called TAP. Okay, transporter associated protein. This one is transporting our kind of a peptide to the endoplasmic reticulum, then through Golgi apparatus, etc. Okay. So I'm asking you, what is tap transport score? We have plenty of tools in IEDB in Raghava. In one tool of Raghava, we can able to calculate tap transport score. Okay, I'm going to freeze soon. Try. Just 5 seconds left out. The choices provided are cleavage efficiency at the C terminus of the peptide. The likelihood of a peptide being transported from the cytosol into the endoplasmic reticulum. The next, binding affinity directly to the T cell receptor. The last option, folding stability of the MHC heavy chain. Very good. I think I can close the poll now. So everyone answer to adhere to a common answer. That answer, whatever answer is right. Okay. I think now we understand some immunology basics. Now let me go ahead. I think even one participant has few questions related to her research. We have time, okay, so I can give last 5 to 10 minutes time to clear your doubts. Any other participants have any other doubts other than a participant who is doing research on bamboo peptides? Anyone have doubts? If no doubts in your research, okay, so I just dedicate 10 minutes to her for clarifying the doubts. Now let me go ahead with the V cell epitope based vaccine design. So here, if you see, we today will be discussing on B cell epitope biology and what are the prediction tools for B cell epitope in IEDB and what are the types of B cell epitope prediction. The first type is the linear sequence based epitope prediction. Number two is the discontinuous 3D structure based epitope prediction methods. And we see computational antibody design. Okay, specifically antigen antibody structure modeling, antibody protein talking. Why I'm taking you this, actually, whatever that participant asked me a few questions related to research, that is related to what I'm going to teach it today. So maintain patience and learn something, and then if the doubts have to be clarified, the last 5 10 minutes we spend. Now let me go ahead with this first website link from Raghava. So actually, if you see, this is an exclusive tools only for B cell epitope prediction. Whatever we have seen earlier yesterday, we have seen so many combination of tools. Like even for proteasomal degradation, we have a different tool. For a tap score, we have a different tool. Okay. Similarly, here for B cell epitope prediction, especially linear, this is one type, and the other is conformational. Under this, we have nearly 1, 2, 3, 4, 5, 6. I mean, we have around 6 different algorithms for predicting B cell epitope prediction. Okay, so BCI-PEP is just collection and compilation of B cell epitopes from literature. BC-PRED from prediction of linear B cell epitopes. How is it done? To predicting through physicochemical properties of the peptides. And ABC-PRED, okay, this is mapping B cell epitopes using ANN. CB-TOPE, conformational B cell prediction method. Okay, so if you want to go with linear, the best is to go with the BC thread. And if you want to go with the next type, okay, structure-based epitope, then we can go with CB2. Okay, we also have other kind of machine learning algorithms to predict other kind of epitopes. One such, of course, something distinguished tool is identifying a B cell epitopes in an antigen for inducing specific class of antibodies. You know that B cell produces types of antibodies, 5 classes of antibodies. Okay, so related to that, what antibodies we are producing? So this IG-PRED is going to predict. Not if all the tools are there in IEDB. Few tools exclusively are developed by our Indian scientists to work on B cell epitope prediction. And now, if you see further, we also have exclusive tools developed by him. I mean, by him and his researchers related to innate immunity and other miscellaneous servers. So, for DNA-based vaccine, we have vaccine DA. For computer design of peptide-based vaccine adjuvants, we have vaccine PAD. So we have plenty of things, okay, including for haptons and other vaccines, cancer vaccines. We have types of tools separately developed for that. Now let me take you to the IEDB. In IEDB, which is the largest immune epitope database, okay, here you can see a separate link called B cell epitope prediction. So here we can predict linear B cell epitopes through this tool, and for the discontinuous or conformational epitopes, you can predict through discotope and elyprope. B cell epitope, just a recap. We also discussed day before yesterday that we have two types of B cell epitopes. If one is linear, we need just the sequence of the epitope. Here we need just the sequence. Okay, if you know the sequence of the epitope, okay, we can come to know about the binding affinity of epitope with the paratopic region. Whatever region you can see that epitope of an antigen is binding in the surface of antibody is what we call it as paratopic region. Am I audible to everyone? Yes or no?
Yes, sir.
Thank you. So if this is a paratopic region of antibody, similarly, if you see the second classification of epitope and paratope, here you can see a part of an amino acid. I mean, few amino acids are getting inside the surface. If it is getting inside the surface, then we can find it's more tight binding when compared to the just having a sequence. So just because we have a sequence of continuous residues, we find like this, and we call this as continuous residues linear epitope. But here, few amino acids have to go through that loops. Okay, go through that per specific conformation. Then obviously can find discontinuous amino acid residues, also called as conformational epitope. In this case, instead of having an epitope sequence, we need a epitopic structure. If only if you have a structure based on the specific folding of the epitope, one can able to see how much effectively it can bind with the paratopic region of antibody. Okay, epitope structure is needed. Here, epitope sequence is needed, which is an input actually. Okay. Now let me go ahead further with a few practical examples. Then only you can understand better. Even we have differences in structure of B cell receptor and T cell receptor. You know the T cell receptor, whatever we see here as alpha and beta thing, okay, it will be like this. And if you see the CD3, okay, whatever you see the CD3, okay, we have two subunits, isn't it? That is actually surrounded by all the four sides. So it will be like this. So CD3 is shown in blue color, and whatever the T cell receptor, okay, I've shown is in red color. This is actually T cell receptor. So actually, this is how we can find in the side view we can find like this, but is in the top view we can find like this. Okay, so in B cell receptor something different, isn't it? It looks like perfectly like an antibody. How an antibody have heavy fragments and light fragments. Generally, whatever we find this red color shown thing, okay, this is generally we call it as a heavy fragment. And whatever shown this green color at the outside, that's what we call it as light fragment or light chains. Okay, so the B cell receptor, which is present in B cell, is just quite equivalent to an antibody. Now related to some information on 3D structure of antibody and antigen complexes. Okay, so if you see this, when to use the epitope prediction methods? Why we have to go for B cell epitope prediction? So the first answer I'm providing is when verified thoroughly that no information is available in the EDB on the antigen of your interest. Whatever antigen you want to search, if you want to search the antigen in IEDB, you can search here. Okay, like there are assays actually. Here we have MHCs. So whatever organism you want to give, we can provide here, and whatever antigen you want to provide, we can provide here. If that specific antigen is not posted, like for example, if Nipah virus W is there, type Nipah here, and you can type here W. And if you find accordingly, if you find a Nipah virus W epitopes, then there is no need to go with the B cell epitope prediction. But if these are not present, okay, then obviously we have to get inside this, okay, that is getting through the B cell epitope prediction tool. Go with either linear or conformational epitope, and then you can predict it. Okay, so this is the first thing, and the second thing is determine the candidate antigenic determinants in an antigen of your interest other than the epitopes provided in the IEDB. So other than the IEDB, if you want to predict few more epitopes and you want to confirm whether any of the newly predicted epitopes can also show better kind of a docking with human MHC molecule, it's better, isn't it? So for this purpose also we can try. Okay, now let me go ahead with the example. So if you download this PDB molecule, so the PDB ID shown is 1808. So you can see that influenza virus hemagglutinin. This is complex with human antibody. This was complex with neutralizing human antibody. Okay. So if you download this protein structure, I just opened this in the Discovery Studio Visualizer. So if I open in the Discovery Studio Visualizer, I can able to see hemagglutinin chain A separately, chain B separately. So this is binding with the paratopic region of the human antibody. Here in human antibody, we have light chain antibody. How you have just now I shown, isn't it? We have an antibody like this, and attached to this, we have light chain fragments. Okay, this is how an antibody looks like. Like it looks like a Y shaped. Okay, so this Y-shaped kind of an antibody is binding with the viral hemagglutinin, that is a surface protein of the virus. So why I'm showing you this is like if you want to check the epitopic region or paratopic region, the generally you know that paratopic region is going to bind with the epitopic region. Wherever it's going to bind here, that is nothing but an epitopic region. Then if you want to find the epitopic binding sites, okay, you can take this protein, you split into two different proteins. You split the light and right chain antibodies in one PDB file, and this hemagglutinin chain A and chain B can be separated into another PDB file. So it can do protein docking. If you do the protein docking, you can able to find the binding sites. So whatever binding sites you find here in the human antibody, that's what paratopic sites. And whatever sites it's going to bind here is the epitopic sites. So like this, again, for example, I'm telling, if you want to try, you download this antigen antibody structure, separate it, and then you can also dock with some online protein docking tools, find the binding sites. And whatever epitope you are going to predict from the hemagglutinin, okay, you can try with the epitopes, whether the epitopes is going to bind with the exactly same binding sites of the human antibody. Got it? You know, this uh, we have to dock exactly, isn't it? So if you want to check this kind of a docking efficiency, okay, instead of targeting protein, we can predict peptide from the hemagglutinin, and then the epitopic peptides we try to dock with the protein, check whether it will get the same binding sites. Okay, this is a cross-check study. Okay, whether the antigen like is going to bind with the even peptide antigen is going to bind with a specific antibody or not. This is another example. Native sperm whale myoglobin. Here you see if you download this PDB ID, you can see this epitope is perfectly binding inside that of a protein. It means that we find a conformational epitope. Unless you have a 3D structure of the epitope, you don't know how effectively it's going to bind with the protein. Okay, so for this, you can check from this PDB ID. B cell epitope prediction, as I told you, if and if you go through these tools, okay, these tools, one can able to know, okay, what are the newly predicted epitopes for the particular target protein of a virus or bacteria. Okay, and I told you an example also that in Nipah virus, I've you published only T cell epitope prediction. The reason why I didn't publish on B cell is already we have multiple journal publications during that time. But for Nipah virus, the best epitope, if you compare T and B, the B cell holds the best for bringing a vaccine for prevention. But for viruses like SARS-CoV-2, T cell epitope is best. So it depends on virus to virus. Now related to linear epitope prediction. So in linear epitope prediction, we generally use machine learning algorithms for predicting it. Okay, and here the example shown is the random forest, which is using B epitope prediction 2.0. In BAP pred 2.0, which is one of the algorithm, which is one of the method to predict B cell epitopes, uses this RF, random forest algorithm. And we also find ABC pred tool. ABC pred tool, all these tools, whatever I showed here, are present in IEDB, Immune Epitope Database. So these are tools actually, and these are the machine learning algorithms, ANN, SVM, etc. So this is how we can able to find these are the algorithms for predicting linear epitopes of B cell. Linear epitope prediction, if you see further, what else we can find. So generally, the linear epitopes are predicted only based on amino acid physicochemical properties, no doubt in that. But here we have subtypes. Okay, the tools uses different algorithms and different logic behind. For example, if you see Chow and Fastman has predicted a kind of an algorithm to predict epitopes based on beta tones, and Emini proposed a surface accessibility algorithm for predicting B cell epitopes. All these are B cell epitope predictions. So beta turn Chow-Fastman, surface accessibility Emini for flexibility, using the flexibility of epitopes. If you can able to predict, then Karplus and Skulls has done this, and Polasker and Tonkoy and Kerr has predicted the epitopes based on antigenic propensity, that is occurrence of residues in epitopes. The fifth is from Parker hydrophilicity parameters. Discontinuous epitope. This is what conformational epitope. So here in IEDB, we have Discotope, Pro, and we also have other kind of an tool from other servers. This uses geometrical properties combined with amino acid scales. The next is geometrical properties and amino acid scales used as an input to machine learning approaches. For this, we can use this tool. We can also use protein-protein docking algorithms. That's how I mentioned an antibody, human antibody, neutralizing human antibody binding with hemagglutinin of virus. Okay. So first try to dock protein-protein, find the binding sites, and try to check whether your epitopes, whatever predicted from the protein, also bind with the same binding sites. Chow and Fastman. This is the first linear epitope prediction. So we're going to discuss, excuse me, this is predicted based on beta turns. You know, in protein structure, levels of protein structure, we have types of uh, secondary structure. The predominant one is nothing but alpha helix. And the next to that, you can find the beta sheets or beta structures, beta sheets. If alpha helix and beta sheets contribute much of the secondary structures, Chow Fashman has proposed that whatever, that is the amino acids like alanine, the single letter I'm writing, glutamate. Glutamate is given as, and leucine, methionine. These amino acids, these are identified as helix formers, while proline and glycine are helix breakers, alpha helix breakers, and these are alpha helix formers. Okay, so according to the Chow-Fastman method, you can able to see the probability that each individual amino acid will appear in a helix strand or turn. So if you see the glycine, where is the glycine, which is the helix breaker, the score is maximum. If you see the proline, which is also the helix breaker, alpha helix breaker, the score is maximum. Similarly, asparagine. Asparagine also has a maximum score 1.56. It means that these amino acids are not going to favor alpha helix. Then it's going to favor next to the alpha helix. You can find beta sheets. So you can find these amino acids in sequence can contribute to the beta sheets. Higher the score, more favorable to the beta sheets. Lower the score, lower the score. For example, isoleucine, okay, phenylalanine. This can contribute to what? Alanine. This contribute to alpha helix. So got it. Beta sheets are shown in higher values and alpha helix are shown in lower values. This is a kind of amino acid scale, and Chow-Fasman predicts based on beta tones. So we only the highest values are considered. Now let me take it to the second thing is the amine surface accessibility scale. According to this scale, all the 20 amino acids have values, and based on surface access, okay, like whatever scores or much higher, for example, arginine, okay, has a high good of surface accessibility. On the antigen surface, you can find much of arginine, like that. Okay, so next to that, we see the corpus and skulls flexibility scale. Sometimes the epitopes flexibility can give a pave a way to effectively bind with the paratopic region of proteins, antibodies. Then flexibility is most important, isn't it? So even for flexibility, for all the 20 amino acids, we have a skill. Kolaskar and Tongoenker antigenicity scale. Okay, so based on antigenic properties, whether whatever protein peptides we have determined has an antigenic properties or not, that is, it's going to give good immunity or not. So that can be measured to the Kolaskar and Tomgetter. So for all the 20 amino acids, we have the antigenicity scale. Next to this, we'll be discussing something on B cell epitope prediction tools. We have plenty of tools for predicting B cell epitopes, other than whatever we have seen in Raghava web server. In linear B cell epitope, we have people, PapiPred, ABCPred, LB2, BCPreds, SVM trip. Similarly, for the conformational B cell epitope, we have plenty of tools. So if you are going for a publication, I suggest you to go with multiple tools, at least 3 or 4 tools, and you compare with each other. Take top 20 and you find which top 20 epitopes have a more frequent occurrence. Based on this, you can finalize it. Got it? So the basal epitope prediction tools you can able to find from the URLs mentioned here, and the methods are also shown here. So related to this, you can find plenty of tools and everything in immunoinformatics journal papers. This is taken from a journal paper. IEDB B cell epitope prediction. So if you see the IEDB B cell epitope prediction, if you go to the IEDB, we can able to find the B cell epitope prediction. Here you can able to see different tools. Okay, predicting linear epitopes, we have a tool for discotope, elipro. This is for conformational epitopes. And similarly, for modeling and docking of protein, we have a separate tool. So it's again these three linked with the conformational epitopes. This is something linked with the linear epitopes. Okay, so not only we have B cell epitope prediction, we also have some other tools like structured tools, lymphocyte receptor automated modeling. For example, it predicts structures of either T cell receptor, B cell receptor, and even complementary determining regions. Okay, so these are selected and we predict accordingly. Lyra, lymphocyte receptor automated modeling. Similarly, structured complexes of epitope receptor. These are also, we have other additional tools, okay, other than B cell epitope for predicting the epitopes. Now let me take you to the first linear B cell epitope. If you take a linear B cell epitope, you have to give either Swiss prot ID or uniprot ID. Okay, in uniprot we have two types of IDs. One is from Swiss prot, the other is from TREMBL. So these together, Swiss prot and TREMBL together or merged together in UniProt. So whatever we find this is a uniprot idea. For example, they provided. If you go to this antibody epitope prediction, that is through this URL, it asks you to type the uniprot ID. Or for example, from beginning we are checking the Nipah virus W protein. So just type the sequence, this ID, or just paste the format, just the protein sequence format, not the FASTA format. Only the amino acids alone you have to paste, and simply have to select any of these methods and submit. Okay, BabyPrед linear epitope prediction 2.0. So this is first tool, and second tool, Chauffasman third, AMNI surface accessibility 4, Carplus and skull flexibility 5, Poloscar and Toengenker antigenicity 6, Parker hydrophilicity 7. All these 7 tools are present in IEDB B cell epitope prediction. There is also so like how we are predicting it. Simply I'm showing you. So after providing the PDB, I mean Uniprot ID, we can get this kind of a result from PepiPred 2.0. According to this, we got so many epitopic peaks. You know why we got so many epitopic peaks at the top shown in yellow color and at the bottom green? Because we have fixed a threshold. The threshold I've given is zero point 0.5. So under zero point 0.5, you can see the sharp line. And in this line, you can see whatever is above the zero point 0.5 is shown in yellow color, and whatever in the bottom is the green color. But if I want to find the best top 10 epitopes, 20 epitopes, what I can do, instead of having a threshold like zero point 0.5, I increase the threshold to zero point 0.6. Then what it can do, these peaks are eliminated. These peaks are eliminated. Only the top peaks alone are taken in consideration. Instead of taking top 10 to 20 peaks, if I want to take top 5 peaks, then I can increase threshold further. This is like a k-fold kind of a method in machine learning algorithm. So this is the top peak, no doubt. So this is the best epitope of T cell, whatever predicted. And next to that, you can find this, I believe, this and this are obviously same peak. So second can be any of this. Now let me take you further. If you see, not only we will be getting a graphical output, but we also get a kind of a table output, like whatever seen in this predictor is this course. Here you can able to see we are around 450 amino acids are there according to this. Whatever I pasted around 450 amino acids, okay, exactly the epitopic region. Initially I kept the threshold as zero point 0.5, isn't it? Okay, only from the 20th position, we can see that the assignment given is E. E means epitope. See here, according to the figure, so up to 20, you don't have any peak, but after 20 can be see the peak is formed. Got it? So this is how whatever we infer from the graphics, that's what you can find in the table corresponding output. So 1 to 19 amino acids, 0 to 19 amino acids are not epitopic peptides, but after 20, you find the epitopes. But if you increase the threshold further, like up to zero point 0.6, then it takes only the peaks present above zero point 0.6 alone will be shown as epitopes. Automatically, the E will be changed accordingly. Got it? Now related to Discord code. I think in between you got the Discord code. Okay. In Discord, we have two different algorithms. Discord top 1 and Discord top 2. If you see this, okay, this is already trained, whatever tool developed by the scientists, these are trained for the 75 X-ray structures. So defaultly, when they tried with the 75 different proteins, they can able to see the area under the curve is shown maximum for Discord top. Maximum the value of area under the curve, more precision. So whatever you predict the epitopes are more precise in Discord 2 algorithm, which is recently developed when compared to Discord 1. So like this, Even whatever linear epitope prediction we have done, Bepipred, Bepipred default it is 1.0. Bepipred 2 is a recent advanced algorithm. Here also I can see the area under the curve is more. So this is a best algorithm can be used. Similarly here, Discotope 2 is the best one. Okay. Now, if you see how the conformational epitopes are predicted, these are using this Parker cytoplastic scale, amino acid occurrence, number of contacts within 10 angstrom, area of relevant relative solvent accessibility. So based on these four parameters, the conformational epitopes are predicted. Discotope, how to work with how to predict conformational epitopes. For this, instead of typing the Nipah virus protein sequence, you have to give a four-letter PDB ID. For example, like this. Instead of giving W sequence, give W structure. Then you have to compulsory specify the chain. If you have two chains like A, B, so default give chain A. Okay, and click sum. Later we can click sum. So only from the structure of the protein, the conformational epitopes are predicted. For this, go to the URL mentioned below. And the continuation of the linear epitope predictions. In between, we have seen structure based. We can see the continuation. We have already seen baby bread. Now, Chow Fastman. Chow Fastman results still same. The threshold. Default, it fixes a threshold. If you want to increase the threshold value, recalculate. Okay, for example, 1.2. Then whatever above 1.2, that alone will be shown. If you see whatever I have shown you in the BepiPred, uh, the Chow Fastman and Imini surface accessibility, for all this I have given common input, that is Nipah virus W. But what has happened, for the first time when I checked with the BepiPred, the peaks I sharply observed after 20 amino acid residues. But here it changes. Here we got this peak, the top peak around 140. Yeah? So after 140, you can find the short peak. Here you check. Here again, if you see according to the surface accessibility, the short peak, whatever we got, is after 170 or after 170 only we got. Here it is after here it is after 140. Here it is after 170. So each and every parameters can find different epitopes, but which is commonly observed that can be frozen. And similarly, Parker hydrophility have different kind of peak epitopes. Polascut and antigenicity have different epitopes. Okay, after predicting the best epitope, what is the next step we have to do? The final step after predicting the B cell epitopes through the scores, the corresponding scores, after that, what we have to do? We have to take only one or two base epitopes, try to dock. Docking is important, isn't it? Then only can check the affinity of antigen antibody interaction. Can be a peptide antigen antibody interaction. Now, let me take you to the B cell epitope. If it is a T cell epitope, I'm going to ask a question, try to answer. If it is T cell epitope, whatever you predicted. So after predicting the T cell epitope, we have to talk with which molecules? We have to talk, isn't it, with human molecules, human proteins. Do you remember what I mentioned to you in the last session? In the T cell epitopes, we talk with human HLA molecules, HLA or MHC molecules. So the target what we try to talk in T cell epitope prediction is MHC or HLA. If so, a specific allele, you remember? I told you for Kerala, I've taken a specific allele and I dropped the corresponding antigen. That's what I published in Nipah virus journal paper. Similarly, here for B cell epitope prediction, after predicting the B cell epitopes, after screening it, Okay, you have to talk with corresponding pathogen recognition receptors. Pathogen recognition receptors, the examples are shown here. Whatever is shown like TLR1 is toll like receptor 1. Like how we find the tolls in each and every region distinguishing. Okay, how to distinguish the regions, we have a toll, isn't it? Similarly here also. Okay, so like the bacteria has much of lipopolysaccharide on the membrane. So if you are to want to target the bacterial pathogen, better whatever antigen we have screened from B cell epitopes, we have to talk with the toll-like receptor 4. If it is a virus, especially single stranded RNA virus, the target receptors of human molecules are nothing but toll-like receptors 7 and 8. Like this, we can able to choose the targets. Like after predicting, after screening, we have to talk with these protein molecules of human. If you are targeting human, the next is I'm explaining something on CABS talk. Okay, the CABS talk is for talking protein versus peptide. So even one of the participants asked me, sir, I am generating lot of peptide from the bamboo, and what I have to do if I want to target a specific thing. Uh, I don't know what exact application might be an antimicrobial or some other thing. So whatever you have, take that microbe. If it is an antimicrobial, take that microbe, and which is the most important target. You find that if you know the PDB ID, you type the PDB ID or upload that protein structure, whatever your model, then peptide sequence you can paste it, and you just try to click submit. One is online tool to dock peptide and protein for this CABS talk. Okay, it uses a flexible docking approach. We also have plenty of tools for docking peptide protein and protein protein. Okay, if you see haddock, haddock is well known for protein protein docking. Okay, so we have plenty of tools to work with protein protein docking. This is actually for protein protein docking. We also have Rosetta online server. Okay, that URL for accessing this tool is mentioned below. Here, if you see ligand docking with a protein, you can try with here. Okay, this is the link one can click. If you want to calculate pK value, you can use this link. Peptide protein docking, flex pepper dock. Okay, you have to click this link and you can input peptide and protein. Similarly, RNA redesign. Docking protein protein, docking 2. Symmetric molecule docking based on type of receptor docking. You can go with the symmetric docking. And energy minimization calculation, energy breakdown. So we have plenty of tools in immunoinformatics, like trying with the even the participant has mentioned something related to alpha fold. We have predicted a structure, how to make it. Use the alpha red to talk with the protein versus protein, and then the binding acids can be this sites can binding site amino acid residues can be determined. Check with flex talk later. Rosetta covalent peptide talk protocol. Simply, if you go to this URL shown, you will go to this. Okay, simply click this link. Then you have to simply paste, peptide and protein. Okay, I've completed my session. Can I go ahead with the practical sessions? So we have just 15 minutes to complete the session. Give me a few time. So as usual, we have to go to the IEDB. So am I audible to everyone? Yes.
Yes, sir.
Thank you. IEDB. So we have to go to the IEDB, and then after going to the IEDB, then go to the basal epitope sequence prediction. Then we have to paste an epivirus W protein sequence. Uh, the protein sequence we have to type from the Uniprot. Uniprot P061, I forgot. So if you want to collect the same thing, it's very difficult if you epivirus. Nipah virus W protein. Let us check whether we can get. Yes, I got P0C1C7. So P0C1C7. Uh, no, I can't able to make it. Better I get inside and take the sequence. So for structure also we have to get from this one. We have a structure here. For conformational epitope, we have to take the structure. But presently, for linear epitope, we have to put the sequence download. So take the sequence. If it accepts the FASTA, I can provide. Even it accepts FASTA, no problem. Give the protein sequence. Let us check what's going to happen. For the PSL epitope prediction, uh, here uh, we can simply click run. Similarly, I try with the structure based. In the structure based, I have to provide only PDB as the ID as the input. So go to the Uniprot structure for Nipavirus W. Do we have any structures here? I'm checking inside. We have PDB ID, fortunately, but the entire sequence, we don't have. We have only the specific binding site amino acid residues. But even it simplifies your task. But if you want the entire protein to be uploaded, then we have to download some model file. Swiss model file is already available here. Okay, and other modeling tools are also available. So for our example, I simply take this one. Okay, 6BVV. Okay, that I'm going to give it as an input for the PDB ID. So go to the PDB ID, type here. Okay, chain A, only one chain. Whether we can check how many chains are available. We have chain B. That's what mentioned here. See, chain B is mentioned. So you must correctly specify the chain, whatever is mentioned. Chain B. Defaultly, it is in Discotope. If you want to try with the ELIPro, select the ELIPro. Discotope 3.0 recently developed. So I'm just whatever is the present. I retain the same, clicking run. So it's getting predicted for epitope, the conformational epitope here. Linear epitope prediction is happened here. Yes, we already got the result for linear epitope. Simply have to click download all rows CSV file. Whatever we are predicting, in desktop, go to the CADVD. So our file is actually related to B cell linear, isn't it? Nipah virus W. So this is how I'm going to save that file. So after this, I simply double click and open it. Okay, whatever we predicted. So zooming in. Now I simply use the wrap text. Wrap text. Okay, so can able to see peptide score. This is the most important one. We have to find the epitopes. You can see the positions are mentioned, but which is maximum? Can we see accordingly? Mm. If I want to predict the epitopes, here we can see only the sequence wise. So it's very difficult actually to check with the table data. I suggest to go with a graphical kind of an output instead of table form. So can we check with the graphical output how we can get? Is the table sequence plots. Sequence plots, but is it related to our sequence? So for each and every epitope, we are getting this kind of a thing. Sequence 1, sequence 2, 0.28, 0.30. The threshold fixed is 0.151. but which is showing the highest peak, we have to check one by one. This is how now presently it is. Okay, point above 0.26. Now I go with above 0.28 we are getting in sequence 3. Again 0.28. So sequence 3 and sequence 4 you find the maximum peaks. Even sequence 5. Generally the P cell epitopes are around 9 amino acid in length. So for the 9 amino acids alone we can check later. 0.8. I think this is the maximum and compared to the other things. In this peak, this peak, whatever you find here, okay, can find the maximum near to the 20. Okay, you can find much of the amino acid residues are following this sequence 6, and this is sequence 7, like this you can see. Earlier, whatever we tried with the pipette, we have options to select them. Even this is also maximum 0.8, sequence 7. So like this, we can able to find from the graphical charts. Okay. The other outputs are in table format, one in the graphics. Or if you want to check from the table data how to predict the maximum value, you can select this, okay, go to the data, filter, okay, and then you can see the A to Z. Again, I think Z to A gives the maximum. See, the positions like 27, 28, 29. Okay, these positions take continuous 9 positions. For example, 27 plus 9, how much? 36. So 27 to 36 has a good pipette score. So if you ask me which is the best one, which is showing the maximum score according to pipette, I can tell you for 9 amino acid peptide, this is the best, 27 to 36. We should have a continuous space. Even though we have 30, 31 is missing, it comes around at least point 0.2 and above. So it means that average if you take, this is the best score. So even in table form, we can also find easily like this. Now let me take you to the P cell structure input. Still it's running. We are yet to receive. But we are, I think one warning is there. Unless it gets stopped, we don't know whether we are getting a result or not. Even the model file you can upload and you can check for the P cell conformational epitopic prediction. Okay, mm, that's it. And now the session is to the participant who want to clarify her doubt. Do you have any doubts in your research? You can now discuss and you can clarify now.
Hi, sir. Good evening.
Good evening.
Am I audible, sir?
Yes, you are audible. You like to share your screen or no need?
Oh, no, sir. I just want to talk.
Yeah, yeah,
Uh, like,
please.
uh, so actually, my research is on functional genomics and proteomics, sir, where using the RNA sequencing, we have identified the peptides which is highly expressed in the growth zones like the cell division, elongation, and maturation zone of the bamboo shoots. Recently I work on the peptides to validate the function of the functional validation of the peptide. From that, I have taken one highly expressed peptide, and when I have cloned and found the function as it suppress the cell elongation in the when I have transformed the peptide into the model plant. Followed by that, as I have completed the functional validation, my supervisor he advised me to predict the structure of the peptide. Also, he advised me to identify the receptor which is responsible, because he said I should find the molecular mechanism underlying this. Underlying this. What to say?
Bamboo should
The
growth.
shoot
Should growth.
growth.
Mm.
Yes. In order to that, I have predicted the peptide structure using the collab alpha fold, sir. And also, as you said, I have done the Ramachandran plotting using ProCheck and found that 76% of accuracy. But he said, predicting the structure is fine, but he needs the perfect validation for the peptide. prediction, sir, so
Mm.
that I will get my, I mean, it will be useful for the publication. Also, he asked me to identify the receptor because my peptide is a novel peptide, and there is no any research which is prior done in bamboo. There are some research which have undergone in the model plants and commercial plants, but though we don't have any proper identification method for this peptide to identify the peptide. So
That's
using the I mean amino acid length and using the transcriptome data, we have identified this peptide. Based on the amino acid length, we have classified it. So I just wanted to know if you have any proper pipeline for the publication, like for
yeah.
prediction, validation, and also for the
Yeah.
receptor identifications.
May I know from which university you belong?
Nanjing,
University
actually, I'm doing PhD in China, sir.
China.
Nanjing
Oh,
Forest University. Yeah.
oh, you are Sri Devi, Madam referee. Am I right?
Yes, yes, sir.
Okay, okay, okay. Yeah, yeah. Actually, even a few more countries also shown interest to join this course, including from Pakistan and other countries, neighboring countries. If you see here, whatever you mentioned related to the peptides, the China is very famous for doing research in peptides. So only even the type of research what you are focusing is also related to the peptides.
Yes, sir.
So China, we have so plenty of web servers for the peptide related check peptide properties, checking peptide designs, etc. But fortunately, these kind of common servers, like whatever is shown,
Mm,
rosie.graylab, jhu.edu, we have
mm.
beta peptide design. The problem behind in peptide is if peptides form beta turns, okay, then
Yes, sir.
the stability is getting lost. Like however you mentioned, if you want to validate the quality of the peptide, okay, whether the peptide is going to correctly undergo the mechanism like increasing the shoot length of the bamboo or not, okay, we have to predict with few properties like however you asked. So you can try with this beta peptide design, of course. Whatever peptides you have screened, you have to check through this, okay, to check these
Okay, sir.
properties, whether it has that kind of a beta fold or not. And then you can check with the whatever peptides you have predicted, even if it is having immune properties or not. Okay, for this you can able
Mm,
to check with like even antigenic properties can be predicted, mutagenic properties of the peptide can be predicted. We have separate tools for it. Okay,
okay, sir.
so that also if required for plant related thing, you can check with it. Toxicity of the peptide, that's also important. Toxicity we can check here. Okay, talk
Okay, sir.
stock. So we also yesterday also I showed you specific tool to predict toxicity for the peptides.
Yes,
Okay,
yes, sir.
pep talks or something like that. You can go through that and you can find it. And for the protein structure, for the shoot length, definitely we have literatures and we can find which is the most significant protein to improve the shoot length. So whatever you find that protein target, okay, you have to download the
Mm,
PDBAD or model it, and whatever mentioned 70 plus, okay, it's okay.
mm.
But because if you don't find any other proteins, you got only that specific protein which is giving only up to not more than 70%. It's still okay, but uh, you can able to carry forward because few publications I have seen even above 50%. That is Ramachandran favored residues. Even if it comes above 50%, they are taking for the publication. Okay, but you check your best, like whether you can get few more kind of proteins or not. If you didn't get, and if I mean, if the model structures even in Swiss doc, you can get only two best models. If you change the template, okay, you may get a few more models. You can also try like whether that models whatever you have received from the changing the template can give a better Ramachandran favored residues or not. Even if it not work successfully, I suggest you to go with iTaser instead of Swiss modeler for homology modeling. because the prediction can be improvised if you go with a threading model. Okay, so like this,
Oh,
there
okay,
are
sir.
plenty of ways. There are plenty of ways to change, what kind of model can be chosen. Whether we can change the templates in Swiss model, or you can change the to like ITSF through threading. Okay, this can give you to improve the Ramachandran plot score.
Okay, so actually my professor, he's not accepting if I say I get 70 percentage of accuracy, because he says when I have analyzed the amino acid sequence in the ESPER-C tool, that in that it shows the
No,
unstable
yeah, yeah, yeah,
peptide. So
yeah,
he says
yeah.
it's
No
he,
problem. See,
um,
actually, if so, you can able to change with the templates or change the server. It's possible with you to get even 90% and above. 90% and above, the scientist can accept. 70%, even I suggest my students not to use the protein structure unless they get a structure from X-ray or NMR techniques. Okay, sometimes the computational model structures won't perfectly if the
mm,
accuracy reduces just because of the stereochemical quality reduces. So even I
yeah,
suggest
yeah,
the
yeah.
same, but you try to change these kind of suggestions. This can able to improve your scores.
Okay, so also for the molecular docking, as I have told sir, the bind using the binding affinity, I can pick up the I mean the best receptor, right? So because for the plants we have RLK's receptors, which is responsible for like plant growth and development. Like for example, in my research for the shoot cellular pathways. So in that way, there are certain receptors which is already found but not in bamboo, while in other plants. As I have taken, I mean after I take the structure, and where after docking using the binding affinity, I can predict, I mean I can state that my peptide have a good binding affinity with this receptor. So maybe I mean uncover my molecular mechanism, right? So from that
You
way.
are right. You are right. You are right. So covalent peptide docking, it gives the best
Hmm.
stable binding. You try with this kind of a docking tool. Okay.
Hmm, okay, sir.
So yeah, yeah. So unless you try with the top 10 or top 20 peptides, you can't able to find which is the best. Okay. Like our even for the antigenicity peptide
Hmm.
screening, however I mentioned, we have different algorithms. Okay, so like this, you have to try with few more algorithms and few more tools and compare three or four tools, and then you can come with a kind of overall talking with a tool added like covalent peptide talking, and then compare 10 peptide scores and which is best, that you can declare it as a finalized.
Okay, so actually, they are mainly focused on functional validation. So I have cloned, I mean, there are about 34 peptides which had a high expression, but from that I have cloned 20. In that 20, only this one peptide have a good function in the, function in the model plants, and like it inhibit the elongation. So he asked me to focus on this and further do any other assay
Yeah, yeah,
related to like
yeah.
Western blotting or something.
Yeah, yeah, it is
So,
better. And even for this, we have some computational part to be worked out. Related to this, tomorrow I'll be taking you related to the
okay,
vector design, etc. So also and try to attend tomorrow's session. Okay, it can
okay,
give
okay,
you
sir.
some clarity on that. Okay, but of course, whatever you do in wet lab, it gives a standardized result. But even in computational, it can work somewhat. Okay, that I will be discussing it on tomorrow.
Okay, okay, sir. Thank you so much, sir.
Good. Okay, thank you. So
Thank you,
nice
sir.
interaction from the participant. Any other participants have any other doubts? Like this, if you have doubts, you can ask today, or if the time is extending, we can also discuss it on tomorrow. Any other participant? Presently, you have a doubt. I can clarify for only one participant at present. No? If no, okay, you can even type. If you remember anything, you can type anything in my personal WhatsApp. I keep it in mind, and tomorrow I can dedicate specific time for you for clarifying your doubts. Okay. Okay. Very good. Now our research, now our training is also extending help to the Chinese scientists. Good. Like this, in future, whatever you learn, try to bring in the form of an outcome. Okay. So in the form of publications or in the form of a research. Unless you practice in the form of a research, okay, so you won't get a full beneficial effect from this course. Okay, and keep follow to our phyto nanostruct technologies for the upcoming courses, and also for the products we company we sell the secondary metabolites. Now our company is also expanding to sell around 100 different secondary metabolites. Okay, like how the Chinese are synthesizing any kind of peptides, whatever we give. Similarly, we are also are going to provide the secondary metabolites of very cheaper price. When compared to China, we give one fifth or one fourth of the price. Okay, so whatever strengths we have to utilize it even for our research. If anyone like to do research with the phytochemicals, keep in touch with us. Okay, thank you. Have a great day. Good night to everyone.